And on to our next speaker, Joe Betts-LaCroix from one of our supporters.
All right, how's it going so far? You've you've held out for a very long and amazing conference. So we're Retro Biosciences, and I'm here to report back on the second year at Retro. So we are working to extend healthy human lifespan by 10 years. So last year I gave some specs on numerical analyses of how the company was going. This year some of them have changed. So we're still operating on the lion's share of our rather generous seed funding, and we've added a bunch of new lab space. We've more than doubled in size. We've added a couple new programs, but we're still one team, and we still have approximately, rounding down, 0 bureaucracy. So, man, that was a really amazing talk, Vera. One of the things that I really love about the academic study of aging biology is that aging touches every single tissue and cell in the body, and so the people in aging tend to be these incredible synthetic thinkers that think across lots of different parts of biology, and that makes it really, really exciting and intellectually seductive. And there's this whole area that we could spend our entire lives thinking about. But Retro right now is disciplined in focusing on this last little hop there at the end, taking things that have already shown robust proof of concept in mice and translating them to humans. We have a bias. So here are the programs that we are working on. And the partial reprogramming section in the middle was one program last year, so it kind of metastasized a little bit into 3 different programs. And so I'll just be going through those one at a time, starting with our plasma program. So I think probably, hopefully, most of you have heard of heterochronic parabiosis, where I know it's a mouthful, you connect together old and young animals, and the old ones amazingly become essentially younger. And that's an exciting proof of concept, but what do you do with that? It's a little awkward, I guess, to translate that to people. For those of you who can't afford a blood boy, we have a solution. One of the questions about that is, do you have to keep this person attached to you your entire life? So I don't want to steal his thunder. Vadim's probably here, but he'll be, there you are. Possibly he'll be talking about an experiment they did in their lab this year. It was really exciting, where you put animals together for a bit, and then you separate them again, and does the effect persist? And amazingly, like even 2 months later, if you look at DNA clocks, methylation clocks, old animals are significantly younger afterward, and also you can see like a very significant lifespan extension. So that's great, but how do we deal with the blood boy thing? Is there some way of making these more translatable, these effects? So one of the things that got us excited about working on in the sort of plasma blood modification space is results that came out a few years ago that showed that you don't actually have to transfer all the blood back and forth, but you can just dilute the plasma in the blood of the animal, and a bunch of the effects of parabiosis persist. So that's obviously a big chunk of it is toxic bad things in the old animal that need to be removed. So how do we do that? We take out half the blood of mice and separate the cells from the liquid part, replace the liquid part with a dilutant, and then combine it and put it back in the animal. And it's extremely difficult to do. It's a lot of careful surgery and so on. But it has amazing effects. And one of the most profound effects that we see is on microglia, which in a normal, young, healthy animal are ramified. And they have these processes that extend out. With aging, they become more and more globular and so-called amoeboid in shape, which is the state they take when they're inflamed. These are in the hippocampus in your brain. You don't want them inflamed. So rejuvenation through plasma dilution indeed makes them more ramified again. And we do this, we have a confocal microscope. We look at slices of mouse hippocampus. We find the microglia. We use computer vision to form a digital backbone of the processes from that, and then we count the ramifications with radius. So here's a result from work that we've done in many such experiments over the last year. And you can see the higher they are, the more ramified, basically. And the bottom, the yellow one, is the old control animals, and not very ramified. And then the red ones are the young controls. And then we apply one dose or one intervention of this plasma dilution, and amazingly, the microglia are restored back, essentially indistinguishable from young. This is the same data broken out into individual time points. This is a compilation of at least 3 experiments with, again, the old animals on the bottom in yellow and the young animals in red. And look at the light blue line. That's 29 days after a single intervention. And you see it's basically indistinguishable from the young animals. So, I mean, that's kind of starting to be something that you could imagine being a tractable, like if you're like wanting to work on aging, like some sort of pill or intervention of some kind that you take once a month or once every 2 months. It's harder to forget than something you have to take twice a day. So we also see durable responses of this treatment toward a young direction in other organs as well. More on that probably next year. So moving on to the partial reprogramming, we have a T-cell program. And that's specifically focused on cancer, which is, strictly speaking, an age-related disease. But on the more micro level, there is an application for reprogramming and rejuvenation of T cells that are involved in killing cancer in order to save the lives of huge numbers of people who are presently dying of solid tumors. And so some of the most exciting therapies against cancer are cell therapies, and they're doing fantastic against liquid tumors, such as leukemia. But against solid tumors, they are not doing so well. 2 main problems are that, one, you make a CAR T cell, send it after a solid tumor, and the solid tumor can mutate and evade the antigen specificity of that T cell is looking for. The other is the T cells get there to the tumor, and they try to attack the tumor, and they end up getting exhausted before they finish the job. So for the antigen escape, we're using an established modality that is not yet approved for humans, but it's a matter of much research, using so-called tumor-infiltrating lymphocytes. And so what we do is we have right now like a fresh stream of tumors coming from our hospital collaborator, and we remove the T cells from those tumors that are exposed to the tumor cells themselves, and then we reprogram them in a way that rejuvenates them such that they then have the ability to replicate more and kill cancer. Also, by extracting the T cells that arrive in the tumor and are fighting the cancer, there's a diversity of different antigen types that are on these T cells, making it much, much less likely that the cancer will be able to change one antigen and escape. So if you just want to see a little sneak peek of some of our data, this is showing the rejuvenation step in that TIL workflow, where we have a couple of different gene sets. On the left is a set of exhaustion-related genes in T cells, and you can see that for a couple of replicates there, that after the rejuvenation step, there's a very significant reduction in exhaustion markers. And on the right, the stemness genes, which have to do with it being more earlier in the T cell differentiation cascade, essentially rejuvenated, massively upregulated stemness markers as well. So that's just a sneak peek. If you want to learn more, if you're fascinated by TILs, our head of T cell therapeutics, Simone Vidal, will be presenting a bit over a month from now in Boston at the TIL Therapy Summit, so come on by. So moving on to HSCs, hematopoietic stem cells, a bit of a mouthful, basically blood stem cells. They live in your bone marrow. They make all of your blood, basically. They don't, strictly speaking, make T cells, however, later in life. So it's very complementary that we have these two programs. But they make all of the rest of the blood. They can make T cells, but they don't, because we no longer, after your late teens, have a thymus, this T cell can't mature and go on to be functional and useful. So all of the rest of the blood is an incredibly important organ, possibly makes the most cells of any other organ in the entire body. It touches essentially every cell. It's been shown that mice that have young HSCs engrafted into them are healthier and live longer. Humans who have young donors, this is in the context of cancer, do better than ones that have older donors of HSCs. So young HSCs seem like a really good thing. So we're working on an autologous method, not wanting to spend the rest of our lives, we as longevity researchers who want to be able to make therapies for healthy people. So we're working on an autologous method, which means that your own, it looks like this, your own cells. Take out HSCs, rejuvenate them by partial reprogramming, then expand them. You can see some results of expansion here. It's very difficult to expand HSCs. Actually, it's not that difficult to expand HSCs, but it's difficult to do it and have them retain all of these extremely subtle capabilities that they need in order to differentiate into 3 different lineages and like actually repopulate all of your blood. And I want all of my blood. And then after the expansion, they are reintegrated back into the blood. And so this is the rejuvenation part, and you can see this sort of reciprocal process where they are losing their sort of CD34 as a marker of HSC identity, and that goes down during reprogramming and mostly recovers. A little bit of work to do there still. And inversely, you can see the pluripotency marker, which is like how reprogrammed-ish the cells are becoming, increases during reprogramming and then decreases mostly at the end. So then how do you get these cells back into the body? Excuse me. How do you get these cells back into the body? Unfortunately, you have a bone marrow when you're older that is full of old HSCs, and there's no room for new HSCs. So as I mentioned, we are thinking toward therapeutics that can be used in healthy people to keep them healthy, not specifically in sick people to like try to cure an acute disease, which is what most of medicine does right now. And so they're perfectly fine using horrific things like radiation and busulfan on these people to make space in their bone marrow. Because they're dealing with extremely acute situations. So we've been bringing up in our lab a non-toxic conditioning method, what we call NTC, that makes space in the bone marrow that we would want to use ourselves. And you can see on the left fantastic data showing that it compares with the standard of care for making space in bone marrow. And on the right, you can see that when we take human HSCs and put them in mice that have been such conditioned that indeed they are rebuilding the blood systems of mice, as evidenced by CD45 positive cells. And also the bone marrow seems to be a bit more functional in that the young cells do better, which is evidence that the niche is intact. So moving on to a much longer term program that is aiming at more whole human rejuvenation. The reason we start with these other T cells and HSCs is that they can be removed from the body. It's just amazing that you can take HSCs out of the body and rejuvenate them and put them back in for such a huge and important organ. It's just kind of a lucky break, I'd say. However, it's not particularly convenient to take out most of your other organs and rejuvenate them and put them back in. So for reprogramming to work on these, we have to learn to do it in situ, in vivo. And so this is a longer term program of ours. It has to proceed very carefully, step by step. So we have succeeded in reprogramming the livers of mice. And we chose liver because it's incredibly easy to deliver reprogramming factors to. So here's just a snapshot. It's a UMAP of gene expression on hepatocytes in mouse. And you can see like there's one particular gene set model that demonstrates reprogramming, the degree of reprogramming. And you can see that's high in this particular cluster that is on your right-hand side. And for that same cluster, you can see the hepatocyte identity is another gene set of things like albumin, et cetera, that is sort of what hepatocytes do on a daily basis. And that is starting to decrease. And then on the right-hand side is an RNA-seq aging clock, single cell aging clock that we've developed based on gene expression that is now getting like shockingly accurate thanks to Alex and team at Retro. And so now we use that as a tool across many parts of our biological work. And you can see there's about a 40% reduction in biological age of those hepatocytes that are reprogrammed. So that's just a sneak preview. Moving on to our autophagy program. That is the program for us that, well, it's, I guess autophagy, another mouthful, but it's the process of redigesting broken intracellular components and making them back into usable building blocks again, like amino acids. It's very complex. There's one particular aspect of it that we have a target and a lead compound we call RTR242. And we spent much of last year developing the ability to synthesize this complex molecule against many different candidates that we tried. One is working fantastically now when we've manufactured 10 grams, and we're in the middle of doing a 1.5 kilogram batch, which is sufficient for our phase 1 clinical trial coming up. We increased the solubility by over a factor of 200 by like a bunch of different formulation work, like a lot of formulation work over the last year. And we've now passed non-GLP tox in several species. And then in parallel, we're working on ramifying this molecule, but based on some modifications into some other compounds that we expect to take into different indications in people. And indications are a challenge for the whole aging biology field, I would say. There is this tradition of what we call disease biology, where you go after specific approved diseases and make cures for them based on the proximal causes of those diseases. And then there are aging mechanisms, and there's fortunately often some places where those overlap. And that's what an aging biology-centered company can then bring out its products for their sort of initial debut in what we call proxy indications. In this case, our proxy indication is a particular rare disease. So those are our programs. We last year announced some clinical aspirations. Proud to say that we have completed our first clinical trial, which was in our plasma program for discovery purposes. And in fact, I guess the day before yesterday, we just got the SomaLogic data back. We haven't finished analyzing it yet, but super excited to see the results. We will have approved therapeutics in patients before the end of the decade. Hang on there. So how are we going to do this? We have this sort of intense ethos of pace and a sense of urgency and of like rapid iteration, trying things, making it better, making it better. We have done more experiments in the first half of 2023 than all of 2022 combined. We have been continuously building lab space. We make them out of shipping containers, which is a bit creative, I guess. And so, our job from a facilities perspective is to sort of keep pace with the growth of scientist headcount. And then we ran out of container space and we built an open lab this year. And it's beautiful to us and functioning great. So we get together every year, the entire company, offsite, and sort of reflect on what happened last year, envision what we want to do the next year, and where the science should go, how we want to continue to working together, like how is our culture working, do we need to tweak it? it doesn't really fit our style to go into like a typical hotel and meet in some meeting rooms for some reason. Working at Retro is very intense. So why not meet intense? Sorry. This is Retro. This is who we are. We are hiring a few more people, operators and especially scientists at all levels. We would love to consider you. Please check out our careers page or just email me directly, joe@retro.bio. Happy to take any questions.
Thank you so much, Joe. That was super awesome.
Yeah, thank you very much. You showed very interesting data in heterochronic plasma transfusion in mice. Do you have any preliminary data in humans?
We will know in a little while. So, like for instance, the SomaLogic data is a gigantic pile of data because it's like many thousands of proteins times many people times many time points and different interventions and so on. So I would say that our comp bio team is pretty chatted, but they're not quite pulling together the whole thing in 2 days. But we'll have data for you later. Any others?
Hi, I have a question about your liver reprogramming. It actually is concerning that there is this kind of like loss of cell identity with reprogramming.
Yeah.
And I was curious whether you reprogrammed the whole liver and it's rather that the majority of cells are still fine, but some are too far along this line, or those are the ones which you got somewhat reprogrammed and the other ones were just untouched?
Yeah, so I think it's I don't know how familiar you are with the overall partial reprogramming literature, but there is this universal feature that during so partial reprogramming, full reprogramming, you take cells all the way back to iPSC, and then, if you're an organism, you're dead. But partial reprogramming, you start reprogramming, and then you stop, and the cells relax back to their original adult state, but with a rejuvenated configuration in their epigenome. And so it's like pretty much universal that cell identity kind of gets tuned down a bit, and just stop me if we're out of time, and then returns. Certainly, if you go too far, you're toast. And that is why this is a long-range program, proceeding step by step. It's what it's going to take if we want to rejuvenate many organs in the body to learn to get really good at this. It has to be uniform. So if you have a delivery system that is putting 100 times more reprogramming pressure on some cells than other cells, and some of them go too far down the reprogramming path, and they may never come back and just go haywire and get stuck in Never Land. So there's uniformity is definitely an issue. As you saw in that UMAP, there were some hepatocytes that were responding and some that weren't. And so we're like limiting the doses at levels where we, if there's always going to be a spectrum like this based on the delivery method that we're using right now, and we're constantly improving on our delivery methods. So those mice do fine. Hopefully I answered your question. Probably longer-winded answer than you wanted. Yeah.
I think. Yeah.
We need microphones. We have to
talk in the breaks. Thank you so much, Joe.
Okay,
that was
thanks
really
so much.
Fantastic talk. Amazing.
Retro has the ironic combination of a sense of urgency and impatience and moving really fast and a really long timeline.
Right.
Right now we have a 17-year cash runway.
Wow.
10 years of healthy human lifespan. Addition is like I have a bunch of people complaining to me saying, why so small? Like, I want to live longer than that. Why are you stopping at 10 years? And then I have this other group of people saying, are you crazy? Biology is hard. How could you ever possibly add 10 years to lifespan?